Imagine using a lab technique, not just for research, but as a real-time diagnostic. To tell a bone marrow transplant patient they're relapsing just by looking at the RNA in their individual cells.
Yeah, and that's the massive leap we're diving into today. It's the core mission of this 2017 paper in Nature Communications, massively parallel digital transcriptional profiling of single cells.
Before this, single-cell RNA sequencing, or, Sc-RNA, it was such a bottleneck.
A huge one. I
Wow.
mean, it was incredibly slow. You were either stuck with these plate-based methods, which meant hours of painstaking cell sorting, what people call FACS,
Yeah.
or you had to build your own custom microfluidics.
Which nobody has time for. It just wasn't practical, especially not for a clinic.
Exactly. So the authors here, they basically built a single-cell assembly line, a high-throughput system anyone could use. They call them gel beads in Emulsion, or GEMs.
Okay, so the core idea is this tiny bead inside a droplet of oil.
Right. And the real innovation is what they load onto that bead. Barcoded molecular tags.
Ah, the tags. So that's the secret sauce. I mean, how did they solve that huge problem of giving every single cell a unique identity?
Well, each tag has two key parts. First, there's a 14 base pair cell barcode, which tells you which cell you're looking at.
Okay.
But the critical part is the second tag, a 10 base pair unique molecular identifier, the UMI.
And the UMI is what allows for digital counting, right?
Yes, that's the key. In older methods, if you copy one RNA molecule 20 times, it looks like 20 molecules. You get this amplification
It
bias.
skews the data.
Totally.
Yeah.
But the UMI acts like a digital serial number. So even if you make 20 copies, you only count the original molecule once. It's incredibly accurate.
And the whole system is fast. I read it can process 8 samples at once, and the cell part takes, what, 6 minutes?
About 6 minutes, yeah. So once they had that unique IED system sorted, the next big question was, is it reliable?
Sure. I mean, you have to avoid what they call multiplets, right? Where 2 cells get trapped in one droplet.
That's the first technical hurdle. To test for that, they did something really clever. They just mixed human cells and mouse cells together. It's obviously very easy to tell those apart genetically.
And the result was great. I saw they reported a multiplet rate of just 1.6%. That's incredibly low.
It's fantastic. And maybe even more important, they got a cell capture rate of about 50%.
Wow. 50%.
Yeah, for a lab working with, say, a tiny precious patient sample, getting half your cells back is a complete game changer.
So with the tech validated, they just scaled it up.
Immediately. They profiled 68,000 fresh immune cells -PBMCs from a healthy donor just to show the throughput.
And that huge data set gave them, what, 10 different cell clusters, T cells, NK cells, all the usual suspects.
Right, but also much finer details, like specifically activated cytotoxic T cells. The resolution was just amazing.
Okay, so this brings us to the really big question. You have this massive amount of gene expression data. How do you use it to tell cells apart, not by what they are, a T cell versus a B cell, but by whose they are? Patient versus donor.
That is the million-dollar question. And this is where they introduced a brilliant computational trick.
Okay.
They realized they could find tiny genetic markers, these single nucleotide variants or SNVs, directly from the RNA sequencing reads themselves.
Wait, so they're using the RNA data to figure out the cell's DNA genotype without ever sequencing the DNA.
That's it exactly. It effectively turns the RNA profile into a DNA fingerprint.
And that is the clinical breakthrough. They used this on an AML transplant patient's bone marrow.
Yeah, and the results? They were chilling. The analysis found 2 distinct genotypes in the patient's sample after the transplant.
And the numbers.
Only 13.8% of the cells were from the healthy donor. The other 86.2%, they matched the host, the patient's original cells.
Which unfortunately was consistent with a clinical relapse diagnosis.
It was. It proved this method could be a high-resolution tool to monitor transplant success, to spot that residual disease way earlier and with more detail than anything before it.
So the innovation here really is democratization. This platform, with its software pipeline called Cell Ranger, it just lowered the barrier to entry for everyone. It took single-cell analysis out of specialized labs and made it routine.
Absolutely. And the clinical implication is you suddenly get insights that were just impossible before. For example, they saw specific types of blood precursor cells expanding in AML patients right before a relapse. That could be a predictive marker.
Okay, let's recap the big takeaways. First, this GemCode platform enabled massive parallel Sc-RNA-seq. Profiling tens of thousands of cells became routine.
Second, it was highly efficient, capturing about 50% of cells, and it used UMIs for truly digital accurate counting.
And third, the RNA-derived SNV analysis. That allowed for high-resolution monitoring of donor and host cells in clinical samples. A huge step forward.
There is one small limitation, of course. Since it's a 3' tag assay, you're a bit limited. You can't really capture certain types of RNA or get info on full-length molecules.
A fair point. But the final thought has to be about the foundation this paper laid. This integrated architecture, the bead, the fluidics, the software, it wasn't just about counting genes.
No, it was a blueprint.
It was the blueprint.
Yeah.
It created the single-cell factory floor that everything else has been built on since. From tracking proteins and gene expression at the same time to mapping chromatin, it just changed the game.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.